All right, good morning. It's 11. It's my pleasure to introduce Susana Coelho and Sodai Jaruwatana. Thank you so much for accepting our invitation to give this talks for the functional genomics part of the analysis committee. They will be talking today about principles of the evolution of multicellularity, sexual systems and development in brown algae. Susana is originally from Portugal, but she did her PhD in the UK, then moved to Roscoff in France, and is now at the Max Planck Institute in Tübingen. And I'm also very excited to hear from Sodai, who's working in the lab of Susana. He's originally from Japan. He grew up in Thailand, did his master's in Cambridge, and is now also in Germany. And I'm excited to hear from him more technical aspects of their work. Susana, please, it's up to you.
Okay, thank you. Thank you for the kind introduction. So yes, we just shared in this talk. So I will start giving you just a broad overview about the context of the study that Sodai is going to explain in more detail afterwards. So basically, in our lab, we're interested in brown algae. And I thought that would be interesting to start by explaining what are brown algae, what are seaweeds, brown seaweeds. So actually, the term algae is actually used to is something that actually doesn't exist, because it's used to group together all photosynthetic eukaryotes with the exception of land plants. So algae as a term is a super polyphyletic group that includes organisms from many of the major eukaryotic lineages that are marked in the slide here with a few asterisks. So the broad phylogenetic distribution of these organisms is actually explained by the evolutionary history of their photosynthetic organelles, so their plastids, but I will not go into detail on that today. What I wanted to say is that for most of these lineages, the extant species of algae are unicellular. So this is the diatoms, the phytoplankton that you see in the oceans. So most of the algae are indeed unicellular. But there's been evolution in other lineages. There's been this emergence of very large and complex multicellular algae, and these organisms have really differentiated organs, complex cell types, and so on. So it's really complex multicellularity. And depending on their evolutionary history, and again on their plastids and on their photosynthetic pigments, we usually separate them in three. So the brown algae, I don't know if you can see my pointer. So the brown algae, the green algae, and the red algae. So as you can see in the simplified phylogeny, brown algae are very much separated from green and red algae that are, well, depending on the phylogenies, but maybe monophyletic. But anyway, so the green and the red belong to the Archaeplastidia, but the brown algae are completely independently evolved. So these organisms are extremely important in our oceans. So they form these underwater forests. They are keystone species because they form the basis of many of these ecosystems. And in particular, the brown algae are extremely important because they form this sort of trees, because in the kelp forest, for instance, there are a sort of underwater forests. So very important for carbon fixation and also to provide the basis of the ecosystems in the ocean. On a more fundamental context, these organisms, so macroalgae, are very important to understand a very important transition in evolution. And this is a transition from unicellular to multicellular development. And arguably, only 5 of the phylogenetic lineages of eukaryotes have considered to be really doing this jump from unicellularity to complex multicellularity. And these are the animals, the fungi, the red algae, the plants, so the land plants, and the brown algae. So in each of these 5 groups, multicellularity arose independently. So the comparative analysis of these different groups allows us to potentially understand what are the fundamental mechanisms for this transition, but also the novelties or the new inventions that each of these lineage used to become multicellular. So the brown algae in particular are quite interesting because they have very fast evolution of multicellularity complexity. So it's a relatively young lineage, so between 250 and 400 million years. And during this relatively short evolutionary time, they evolved a fascinating diversity of multicellular forms. The red algae, in contrast, are actually the oldest multicellular lineage. And they have a number of very interesting characteristics. So they don't have flagella, centrioles, the chloroplast, they lack external endoplasmatic reticulum. They have unstacked thylakoids and so on. So very, very interesting organisms with a lot of novelties that we are just starting to explore. So when I mean complex multicellularity, what I mean is really complexity. So for example, and now I'm mainly talking about, I will focus more on brown algae, but what you can see, for example, is that the brown algae have plasmodesmata, which are very important structures for cell-cell communication. These are very similar to the plasmodesmata of plants, of land plants, but these structures evolved independently to the land plants. They have pit connections, they have a number of complexity in terms of morphology of cells, and they have a very tightly regulated developmental program, including very tightly regulated embryogenesis for some of the species, and this will be something that Sodai will mention in a minute. They also have a complication, an extra complication, which is the fact that they have kind of crazy life cycles. So I'll guide you slowly on this life cycle. So at one extreme of these types of life cycles in nature are the diplontic life cycles. So these are the classic ones, the animal ones, where all the development of the organism takes place in the diploid stage of the life cycle. So these are the animal life cycles. And then the gametes are the only representatives of the haploid stage. And then at the other extreme, you have organisms that have all their somatic cell divisions take place in the haploid stage, and the zygote is the only representative of the diploid stage. And then the algae, many macroalgae and seaweeds, brown seaweeds, have these haplodiplontic life cycles where they have complexity and multicellular development in both the haploid and in the diploid stage of the life cycle. So this makes an extra complexity that can be complex, but can also be used in a way to understand very fundamental processes. And actually, this is what we used in the paper that Sodai is going to describe you in a second. So what have we been doing with these organisms? So these organisms have been very well described, actually taxonomically and morphologically. But until about 10 years ago, we had little information about their genomes, transcriptomes, and so on. So when I was still in Roscoff, together with Mark Koch and Akira Peters and many other people from Genoscope, from a big consortium, we started sequencing these genomes for a number of brown algae. So the paper just came out. So we have almost 70 reference genomes of variable qualities, but importantly, they span the whole diversity of the brown algae in terms of phylogeny. And in the meantime, so this was for the genomes. So what I've done in my lab, and this was mainly a project from DRC. So what we've done is to do as this tissue-specific, so atlas of expression. So taking detailed developmental stages across a number of representative brown algae, and we just did all the transcriptomes, and we are complementing the transcriptomes with a number of other omics, DAC-seq, ChIP-seq, and so on. And the idea is to understand the genetic pathways that are important for the multicellular development and complement this with regulation. How are these genomes regulated? And very recently, this is really on the way, we are working also on single cell sequencing, so to understand the evolution of cell identity across the brown algae. But I'm going to stop here because what Sodai is going to tell you about is that we've been using this data from this transcriptomic atlas of expression, which shows a subset of species. And we decided to look at this very old question, which is hourglass patterns of morphology and molecular hourglass patterns. And given that the brown algae are a separate group that evolved independently from animals and plants, our question was, do brown algae also have an hourglass of embryogenesis? And I'm going to stop here. I'm going to give the stage to Sodai now. Stop sharing. Yes.
Okay, yeah, thank you, Susana, for the wonderful introduction, and also everyone for the opportunity to present here. And so, yeah, basically, this is where I come in, right? And before I began my PhD, I had basically no idea of what Susana was talking about just then about brown algae. But yeah, the more I learned about them, the more I came to appreciate what these organisms can tell us about more fundamental principles of evolution and development, or evo-devo. And I think for me, the first striking thing was that they look like plants, right? As Susana said, but they are very unrelated. And the cool thing, again, is that they evolved complex modularity independently. So another fact that really surprised me was that they're more closely related to the oomycetes that caused the Irish potato famine or the plasmodium parasites that causes malaria than to plants. And they have evolved this state independently of animals, fungi, and red algae and plants, right? And to produce the large organisms with multiple distinct cell types organized in this 3D context, in contrast to simple modularity, they evolved the process of modular development independently. And yeah, of course, there must be many lineage-specific features that make each brown algae very unique and interesting. But I was interested in features that are shared with other lineages and to learn how this can inform us about more fundamental principles. And luckily for us, with the File Explorer and Atlas datasets, it's only really now that we have the genomic and transcriptomic tools to focus in on this group. And yeah, and the question I basically wanted to test was the development of hourglass, which is a big question in evo-devo. And just for some context behind this this hypothesis, since the time of Darwin, since like 1800s, it's been observed that certain stages of embryo development look rather similar between embryos across different species than other stages, right? And this is especially when the basic body plan is being laid. And so as people looked at even earlier developmental stages, they observed that the early stages are quite divergent between when you look at embryos across different species. And the interpretation was at the morphological level that you have evolutionary divergence at the early stage, bridged by a mid-embryonic period with evolutionary conservation, and evolutionary divergence again as the organisms or the embryos take on species-specific features. And this phenomenon is called the developmental hourglass. But there is a bit of a catch, and already since 150 years ago, people such as Wilhelm His has noticed that some of the drawings by Haeckel presenting the similar embryos are actually maybe misleading. You can see here that this nose is a bit bigger in this species, for example, and it continues to be debated till today. And so measuring evolutionary divergence and conservation at the morphological level can be difficult, right? And so then people asked, okay, is there a way to overcome this? And one way to look at an embryo is through its gene expression, which can be quantified through RNA-seq. And with this, we can then basically test the hourglass at the molecular or transcriptomic level. And okay, so let's say now we have the RNA-seq data across different developmental stages. Now, how do we measure the divergence and conservation? Well, conceptually, one obvious way to do this is to compare the expression of genes in embryos between species. And in this simplified example, imagine you have your favorite organism, the frog, here, and you sequence the developmental transcriptome. And then you compare it against the developmental transcriptome of let's say, your second favorite organism, the fish. And then with this, you can get a sense based on comparing the expression of orthologous genes through correlation or distance-based approaches of which stage is more conserved at the expression level. It's really simple, but there are some limitations to this approach. So the question arises, what happens when you don't have clear orthology relationships? What to do with genes that are only found in one species and not in another? And how do we actually match the developmental stages precisely between species, especially when comparing across deep evolutionary time? And another approach which sidesteps this issue is an index-based approach. And so again, let's say you have your favorite organism, the frog, you have the developmental transcriptome, and then you measure the contribution of more evolutionarily conserved or more older genes in relation to the more evolutionarily younger genes at each given stage. So let's say in this case, you have in the mRNA pool, a lot more transcripts coming from older genes, younger genes in stage 1, followed by more older genes in stage 2, and the situation reverses in stage 3. So this is kind of what we'd expect with the development of hourglass, and we can say that this whole transcriptome has a younger profile in this stage compared to this stage. And when I began this project, there wasn't a clear bioinformatics software available to infer this gene age, especially in non-model systems in a fast way. So we went on to make one. And so this is our software GenERA, or GeneERA, which allows you to look at the evolutionary age of each gene. So here you have your organism of interest. You sequence the genome, look at the genes, and then you perform pairwise sequence alignment against a taxonomically resolved database such as the NCBI-NR. And we see, for example, that this first gene here is super old. You can find it in all the species representing members of these nodes all the way to the deepest one. And so we can put the relative age of 1 here, let's say. And then for this gene, it is relatively younger because you can't trace homologs after this node. And so the relative age is 3. And this tool is also fast and sensitive using the pairwise sequence aligner Diamond version 2, but we also have options to allow for not sequence similarity searches, but structure similarity searches, the use of search against nucleotide sequences, and so on. So yeah, feel free to use this tool as well. But anyway, we can now obtain the gene age, right, with GenERA. And with it, we can, with the RNA-seq data, obtain an understanding of the transcriptome age. But of course, I'm not the first person to think of this idea. And in fact, 15 years ago, 2 groups looked at embryo development in vertebrates and insects using these approaches. So the transcriptome age approach, as well as this distance-based approach, and found a conserved period of development corresponding to the mid-embryo. So on the left plot, you see on the y-axis the developmental stages. On the y-axis, the transcriptome age index. And you see here that in the mid-embryo, you have a lot more older genes being represented in the transcriptome. And on the right plot, the message is basically the same. Here you have the least expression divergence between closely related species. Anyway, so such patterns have now been observed in many animal systems in both bulk and more recent single-cell studies. And I'm just going to dive a bit more into how the transcriptome age is actually computed. So the transcriptome age index is simply a weighted mean of gene age by its normalized expression level, and it's quite a flexible setup. So for example, this here, the gene age at the phylostratum level values can be replaced with other evolutionary measures of interest, such as the DNDS. And then with this setup, you can easily perform permutation tests to quantify the significance of any reported transcriptome age patterns. And all of this can be done with the R package MyType. But yeah, back to the biology. Animals aren't the only group to evolve complex multicellularity. And so the question became, do we see similar patterns of conserved mid-embryo stages in other lineages at the transcriptomic level? And this has been looked at in plants. So I'll just rotate the axes to make it similar to what we see in the animals. But we see that in the mid stage, you have a lot more older genes being expressed, this torpedo stage of the Arabidopsis embryo. And then this has been followed up in other studies in plants as well. And the molecular hourglass has also been described in fungi, but it's not fully resolved, and it's not really clear what the corresponding process to embryos are in this group. So what about brown algae, right? And to test this question, this is kind of why I come in. So we created an experimental setup to sequence the developmental transcriptomes of several brown algal species, including 2 species of Fucus, which are morphologically complex, so Fucus serratus and Fucus disticus, as well as 1 species of Ectocarpus, which is more morphologically simple. The good thing about this is that they all have external development, so none of the samples are contaminated by maternal tissues, for example. And you can see here that the Ectocarpus life cycle stage is pretty intimidating, but in this more morphologically simple species, you just have to remember that there are 3 unicellular stages that bridge the 2 distinct stages of multicellular development. In contrast to Ectocarpus, Fucus has a more complex morphology with a clearly defined brown algae version of embryogenesis. And they behave basically like animals, where you have female and male gametes fusing, forming diploids and forming diploid adults. And so when we looked at the data in the embryo stages of Fucus, we were surprised to see that there's also a corresponding molecular hourglass in these species in both Fucus serratus and Fucus disticus, where the mid-embryo stages here are a period where a lot more older genes are being expressed in relation to younger genes. And this corresponds, interestingly, to the stage or period where you have the transition from cell differentiation to growth in these species. And not just at the transcriptome age level, but we also see this pattern when comparing whole transcriptomes between species by looking at orthologous genes. And yeah, so we looked at whether the mid-embryo stages were associated with increased expression of older genes or decreased expression of evolution younger genes, and we find that it's the latter being the case. And the mid-embryo was also associated with the enrichment of more broadly expressed genes. And this could be related to potential pleiotropic features of these genes being expressed in the mid-stage, causing some of the constraints. And, with these results, I was just wondering, what is there in experimental evidence in brown algae thus far? And when I looked through the literature, what was quite surprising was that these early stages in the analysis that I did here shows that they're quite young. And we also see this correspondingly when you look at how these algae embryos first polarize. They have so many different modes of polarization, and yet they begin to produce similar multicellular structures. And so with this, I suspect that the hourglass pattern that we see here might, at the transcriptomic level, might really reflect the morphological patterns that is to be tested. And so when we looked outside of embryos into adult Fucus, we found that in the species with separate males and females, that the males generally express a younger transcriptome than the females. And this is especially striking in the reproductive tissues or receptacles and the gametes as well between males and females. And in the species that are cosexual or hermaphroditic, loosely defined here, you see that where there are sexual differences, you see the males expressing a younger transcriptome than the female. And so this is quite interesting as well, because it shows other examples of potentially convergent processes outside of embryo development seen in brown algae, animals, and plants. And so I spoke about all this complex development and many different tissues of the adult Fucus. But when you look at Ectocarpus, I mean, they are morphologically very simple in contrast to Fucus, and they do not undergo the same type of canonical embryogenesis that's observed in the Fucus. Here you see that they consist of filaments in the mature gametophyte stage. And yeah, so they have 3 unicellular life cycle stages. And when we sequenced the developmental transcriptome in Ectocarpus, we found that it's just generally unicellular stages expressing a younger transcriptome than the multicellular stages. And this is not only at the transcriptome age level. When we look at the expression distance as well, we see that unicellular stages here are more distant compared to multicellular stages when we compare Ectocarpus with Fucus. So in both males and females Ectocarpus. And so this creates like a broader image that multicellular development constrains transcriptome evolution in general in Ectocarpus. And so just to summarize a bit, at the transcriptomic level, we provide evidence for the developmental hourglass in Fucus. And on the other hand, in Ectocarpus, which is more developmentally simple, we see that the multicellular development itself constrains the transcriptome evolution. And so, you might be wondering what's going on here. We hypothesize that these 2 patterns are in fact linked. And so in the more simple species, cell differentiation and cell differentiation occurs throughout the entire multicellular phases of the life cycle, as well as cell growth and so on. Whereas in the more complex species, a lot of these processes become concentrated towards a more singular point in the mid-embryo, and the regulatory complexity associated with these processes may constrain the transcriptome evolution. And this could be one of the ways in which the hourglass patterns emerge. And so, yeah, overall, the results here demonstrate at the bigger picture the power of using brown algae in evo-devo research, the novel evolution of complex multicellularity, cell types, and I think there's a lot more to come in this direction. And yeah, this work is now published, and of course, this project would not have been possible without all these wonderful people, with the supervision by Susana on the biology and evo-devo side, and Hike as well as on the computational side, and Min, Remy, and Daniel for sampling and sequencing the transcriptomes. But of course, as you all know, there's a lot more going on behind the paper than what's really there. And I think to kind of summarize this whole challenge, the aspects that make brown algae interesting are basically the same aspects that make them hard to work with. And so I had to overcome some of these challenges. And yeah, I already talked about the interesting biology of the brown algae, Susana as well, with the sexual system, cell types, multicellular development. And another point to note here is that, as in all organisms, there are many lineage-specific genes. But, so what you can see here is that all the genes in the brown algae here will be Fucus being grouped into different evolutionary strata. And so the genes belonging here are the genes that are associated or are only found in brown algae. And here would be genes found across all all cellular organisms, including bacteria. And so for genes with homologs found here, we can kind of infer their functions from studies in animals, plants, yeast, other algae, or straminophiles. Difficult word. However, like when you have these genes around here, a lot of them remain a mystery without further characterization. And this could lead to quite funny cases, or sometimes depressing cases, where a lot of the genes that you're interested in are not annotated functionally in any way. So for example, if you just look at the Go terms, you see here in one of the things I was doing, there was an enrichment for there not being a Go term. So, it's quite hard to interpret what's going on here in terms of the function. And this is the same analysis with interpro scan. It's a lot better, but, it's still like this. And so, yeah, I imagine this to be the case with other non-model systems as well. And so for more traditional analyses that's done with models, organisms, or organisms that are non-models but are closely related to other models, a lot of this is kind of not available in all cases for the brown algae. And so a lot still has to be basically built up from the ground up in brown algae, right? And that's why ectocarpus is such an interesting model. And there's already a lot of interesting things in this direction, as Susana mentioned, with like mutants where you don't have a certain cell type anymore, or like life cycle mutants, sex determination mutants, and so on. But there's still a lot to be done, right? And so, yeah, this lack of genomic information might seem kind of depressing, but it was precisely what motivated the focus of the paper, or what then became this paper on the gene age inference and the transcriptome age index. And what I mean by this is that we can use the lack of information, i.e. the lack of homologs, and this is already interesting in its own right? And by combining evidence from different analysis angles, we then built up a coherent picture about the developmental system. And this required also some creativity, for example, to test whether the broadly expressed potentially pleiotropic genes are associated with the mid-embryo. We created a new metric, the transcriptome specificity index, or transcriptome kind of expression breadth index, which uses the weighted mean of gene expression by the expression specificity. And in this case, I used the tau score, but of course, there are other methods such as the Gini coefficient and so on. And as I also showed you before, we also explored expression similarity between species or distance between species, which supported the data when we used the index-based approaches such as the transcriptome age index. And here, basically, instead of relying on one-to-one orthologs, we analyze the data at the whole orthogroup level, allowing us to capture a lot more genes in more sparsely annotated genomes. And this allowed us to capture the expression data from as much as possible. And so to bring some of these ideas to other users, we also invested some time into software development for this project. For example, some of the new features in MyTy now allow us to easily check the robustness of any type profiles to different data transformations on the expression level as well as at the gene age level, which is quite cool. And you can also now perform permutation tests between two conditions, not just across the whole time points, to test whether the type values are significantly different. And of course, there were bioinformatic challenges, but there are also some non-bioinformatic challenges. And as you can see here, I really have no idea what I'm doing, but I'm just accompanying. Yeah. And so to add some context to the previous conclusion, well, we can learn a lot about non-model systems, but there are, of course, many challenges. I found that the gene age and transcriptome age approaches work quite well. And overall for me, all this stuff was super fun. We used these unique organisms and new technologies to test these really old questions. And so I wonder, as we sequence more of the tree of life at the different omics levels, what other interesting biologies that defined, right? And so, yeah, I thank Susana, Coelho, Min, Jose, Remy, Daniel, and Fabian on the Max Planck side and also Heike and his team for the computational advice and supervision. And this project also heavily relied on huge databases such as SwissProt, NCBI-NR, and so on. And it would not have been possible without the current drive to produce all these reference genomes and annotation data across the tree of life. So thank you to you all as well. All right. Let me know if you have any questions.
Thank you, Sodai and Susana, for that amazing talk. It's incredible how you've collected all these different data sets and developed technical tools to deal with them as well. We have a number of questions on the YouTube channel. The first one comes from Kev, and he's wondering, are the older genes also more conserved within closely related species? Are they likely, do they likely serve more conserved functions?
So that you want to answer or I answer?
Yeah. So for sure, yeah, all the genes are quite conserved, and when we looked at the go terms for the genes implicated in the molecular stage of the life cycle, this was not like the case that what I showed now, where we have enrichment for like processes that's obviously associated with molecular development. Yeah.
So
Did I get the question
yes,
correctly?
yeah, more conserved genes are in older genes have more conserved functions, but of course we cannot guarantee that the same functions are occurring in the brown algae, right?
Yeah.
But
exactly. Like as I mentioned before, even if you have because of a lot of the independent evolution of many of the biological features in the brown algae, let's say you have an enrichment for a gene associated with, I don't know, root growth in plants. Okay, the roots or like rhizoid systems evolved independently in brown algae. So, this is also some stuff that we have to take into account. Yeah.
Okay, and for people not so knowledgeable about the hourglass theory, why would you expect less conserved expression at the early developmental stages?
Susana, do you want to?
say again, because I was looking at the same time, I was looking at the questions next. So the question was about the hourglass.
Yeah, so
why we would expect? Yeah,
Sorry.
this is more historical. So that you want to maybe to answer that. It's more historical, right? There was this old Haeckel, what Sodai was showing, that there is this conservation during the mid-embryogenesis where embryos look more alike between different, like between chicken and mammals. There's this stage where there's more conservation of the morphology. And then more recently, Denis de Boul's work showed that actually there is a molecular conservation also at a certain moment of embryogenesis.
Yeah.
So we just repeated this in a lineage that has independently evolved the embryogenesis, right? But it appears to hold, right? It looks like there's a particular moment, probably when cells are defining their fate, where all genes are necessary. So this could be like transcription factors or things like that. So there seems to be this universal conservation of some events that take place at the moment where you define the cell fates.
Yeah, and also if you think about it naively, right? You would imagine a developmental system to maybe begin from the same starting point or a more conserved starting point and increase in divergence as the embryo takes on species-specific features. But this whole hourglass studies show that you do not have to begin from a very the most conserved transcriptome and everything at stage 1, right? You can begin with a lot of variation in the early stages, and this is shown in studies looking at gene expression variants as well. Yeah.
Yep, interesting. Teresa Manuzaki says, amazing talk. I wonder if the expression can be measured at the pathway level, as multiple genes, especially members of the same family, could act in different species with similar function. For example, trans factors.
I guess you're meaning like gene regulatory networks. If we would do, I'm not sure if this is what is meant with the pathway level. So that what you think, doing gene regulatory networks over?
Yeah, so I did not present the next work that like kind of like the next chapter in this story, but I'm currently working on a project that will look precisely at a lot more this question in a lot more brown algal species with less less focused developmental stages, but rather single cell, multi-cell, some embryos and different tissues, and seeing whether we can build a coherent gene regulatory network defining sporophytes, defining gametophytes, males and females. And yeah, this is definitely what we will look at.
Yeah, and then I think it's transcription factors that is meant, right, in the question.
Yes.
So I guess so. I mean, I can't remember, so that maybe I think there was an enrichment in transcription factor expression at those stages.
Yes, but
yeah, that's what we would expect indeed.
Yeah, but because there were so little number of transcription factors in general that we can predict at that time in brown algae, yeah, it was not so obvious how this plays in. And also when you look at genes that are differentially expressed across development as a whole, a lot of the transcription factors that we predicted, some of them were differentially expressed, some of them were not, and when you have just a set of like 160, it's kind of hard to make a strong claim on this angle. Yeah.
Okay. Robert Waterhouse is asking, how does the taxonomic representation in reference databases impact the quality or resolution of estimating gene ages? Do you need to build a special reference set to avoid biases?
Yeah, so this I did not talk too much about the gene age inference in this talk, but we added some heuristics to avoid potential database contamination, right? And there's also sequencing contamination of your species of interest as well. And you can, of course, make a special reference set from these taxonomically resolved databases to then query them. So with another thing that we added to Genera as well is the homology detection failure. Maybe I don't need to share my slides, but basically the idea, wait, I'll just share my slides again. Wait a second. Yeah, the idea here, for example, can you see my slides? Yeah. So the idea here, for example, is that we can then look at individual homologs with the highest bit scores and then look at whether first it's because of contamination or whether some of the new genes that we observe are really novel lineage-specific genes, or you just don't observe homologs any further due to the fast decay when you look at the bit scores. And so this is still a problem with looking at these questions in these databases, but we are trying to, yeah, resolve them one by one.
I also had a question there about the transcriptome H index. If I read it correctly, you weighted by gene expression.
Yes.
And I don't understand the weighting by gene expression in that formula.
So basically, you normalize the gene expression and wait a second, let me find the formula. Wait, yeah. So you basically, for each gene, you have a value, the estimation of gene age. And these are going from 1 to 15, let's say, with 15 being the younger genes and species-specific genes, and 1 being genes found in other cellular organisms. And you then take, let's say, the TPM of that gene at that given stage as well, and you divided the TPM by the expression value, the total set of genes expressed at that given stage, the expression values. And then this is kind of the way you compute the transcriptome H index. Yeah, sorry if I didn't explain it so well.
Yeah, I guess I'm still a bit confused about why the weighting of the gene expression is important, because you want to look at highly expressed genes and give those a stronger score.
Yeah, that's the idea. And with the transformation functions that we did in the paper and that's added further to MyTai, you can then look at, okay, if I do a square root transformation, log transformation, R log, even like non-parametric rank transformations, will we see the same pattern? And the answer for this for the brown algae is that yes. So it seems to be like a whole transcriptome thing, not just driven by a few highly expressed genes. But this is a way to integrate the expression data into the analysis. Yeah.
Yep. Okay, thank you. Iker Irizarry is asking, how do you think genomic data can help in improving functional annotation of genes with unknown function, for example, gene co-expression networks?
Oh, yeah. So, for example, in the next project I'm working on, if genes are co-expressed with other genes involved in, let's say, the rhizoid identity or receptacle identity, then we have a pool of genes that we can say are associated with these processes. But in terms of genomic data from other lineages, right? So this would be improved functional annotation, basically. Yeah.
Yeah, I mean, I was not sure about the question if it's the functional annotation of the brown algae, right? But in any case, this gives you a hint. But what we are trying to do is to develop reverse genetic tools to go for the function. So doing knockouts and really see the function of these genes. I mean, we don't have the ambition to do it for all genes, but at least for the ones that are, for example, in these gene co-expression networks, if there are some genes that are interesting and we suspect they are related to some specific developmental aspect of the algae that is interesting for us, then we are doing CRISPR-Cas to knock them out and then validate their function. Yeah, but I think we are still far from other mainstream models.
Yeah. But in the study, I saw you used protein-protein interactions to go towards the pleiotropy. So I guess guild by association tools would also be possible to figure out gene functions.
Yes, and I think now we are increasingly using AlphaFold and AlphaFold multimer for some interesting proteins. So this is also helping us looking at interacting proteins, so they will be working in the same pathway, for instance. So,
yeah. And also, it's not just the genic sequences, like the coding sequences that are informative, right? The promoters and so on, if there are also certain motifs that are found for the genes expressed in the same co-expression pathways. And yeah, like we can start to build like a bigger picture about the regulatory network in brown algae. Yeah, so it's a lot of things ahead and it's pretty exciting.
Yeah, thank you. There's one more question on the list from Florian Maumus. Is there convergent evolution or common ancestry of some multicellularity genes between Viridi plantae, for example, Rhodophyta, and brown algae?
Yeah, this is very super cool question. I suspect that will be probably both. We have previous work when I was still in Roscoff. We know that some genes that are important, for example, for transition, life cycle transition, these are common and these are ancestral, and these genes regulate the haploid to diploid transition in green lineage, in algae, in brown algae, and in fungi, for instance. So this is an example of ancestry for this important transition in the life cycle. But then there are other genes that there is convergent evolution. So we showed that this master male determining gene in the brown algae has been independently co-opted in brown algae and in animals for sex determination. And we showed that this was, they were not common. It's not the same ancestral protein that was involved in sex determination. So I think we will find both things. It will be very cool. We are collaborating with people working on plasmodesmata evolution, for instance. So trying to look at, for example, are the proteins involved in plasmodesmata in green lineage are the same proteins involved in plasmodesmata in the brown algae, because the structures are very similar. I don't have the answer for now, but I think we will find probably both. Yeah.
Okay, thank you. I don't know if there's anybody in the Zoom that still has lingering questions. If not, then I can thank you again very much for this fascinating talk. Yeah, it's really a privilege to get to see the behind the scenes of such work. I think Louisa, she will probably edit a little bit the YouTube talk and re-upload it later again, cutting off the end and the start. So then with this, yeah, I guess we can we can go and have lunch.
Okay, well, thank you very much for the invitation.
Yep.
And thank you
Very
as well.
nice meeting you. Thank you.
Yeah. Bye.
Bye.